Hello students, you are in the botany class today at Yup Master and we are going to continue our topic in cell as a basic unit of life. In the previous class we have completed up to the plasma membrane and its function. Now the next topic that we need to study is about the cell wall. I told you that there are certain structures which are common in prokaryotic and eukaryotic cell, plant and animal cell that include cytoplasm, plasma membrane and ribosomes. So far we have completed the one common thing that is called as plasma membrane. Now the next topic or the next thing that we have to start with is cell wall, right? So the cell wall you can see in the members of fungi, plants, also in bacteria, right? So you can see these things also in bacteria. And algae. Okay, here I am saying is like in the members of kingdom fungi, in the members of kingdom plantae, that type of character you can mention. So whenever these things are given, in that cases we say that the members or the kingdoms which are having cell wall, 4 kingdoms: Monera, Protista. Those in Protista which are having the cell wall are obviously unicellular eukaryotic algal members. Fungi, plantae, remaining algae and terrestrial plants. Okay, so you can find the cell wall occurrence in 4 kingdoms which are mentioned over here. Okay, the thing is, the cell wall is not only giving the shape to the cell, it has certain other functions. The cell wall depends, it's usually considered as permeable, but still it is deciding the permeability towards the macromolecules, whether those particles are allowed to enter the cell or not. Next, we say the cell wall, if we are considering the cell to cell interaction, means cell wall is making a communication with the surroundings also. It's not on the basis of the responses. Cell wall is not going to respond the things, but it is like the cell wall is having certain windows or the door like structure which are allowing the exchange of material between the adjacent cells. That will be studied as plasmodesmata. Actually, what Robert Hooke studied, I repeat, what Robert Hooke studied were the cell walls. And if these are living cells, in that cases, these living cells must be interconnected. These are going to be interconnected in that case with their surrounding cells. You can say that entire of the protoplasm is partially divided into cells. Okay, the protoplasm is partially, it seems like the protoplasm is partially divided into cells. Because there is a continuity of protoplasm from one cell to another. So this is the way by which the cell wall is helping in interaction, exchange of substances. These connections are called as plasmodesmata. Remember what I said, in the living cell. In the living cell only, not in the dead cells. If the cells are living, then only they have this type of structure, right? So that is the thing. One more word you need to add here, and that is what? Protoplast, which is obviously not same as protoplasm. What is protoplasm then? This is very clear to us that the entire cell content is mentioned as protoplasm, which can be further divided into cytoplasm and nucleoplasm. But what about protoplast? So we say that bacteria, fungal, algal or plant cell devoid of cell wall. If the cell wall of these cells get removed, if the cell wall of these type of cells, bacteria, fungi, algae, plantae, got removed, Whatever left behind is mentioned as protoplast. So the word protoplast is applicable to plant-like cells, not the animal cells. In animal cell, the content inside and including the plasma membrane is protoplasm. Whereas in these type of example, we use the word protoplast. So if someone is saying that what Robert Hooke observed, so he observed lack of protoplast. Protoplast wasn't there. He observed only cell wall, right? So the protoplast word is applicable to plant cells, not the animal cells. Okay, right? So here is the thing that we need to study further. The cell wall that we are going to start with is present in such type of example as I told you in the kingdom Monera members, means the bacteria. It is present in the members of kingdom fungi, the members which are belonging to algae among the classes or among the kingdoms, the three kingdoms in which algae are kept, and in the plants. In bacteria, the cell wall is made up of peptidoglycan and lipids, most of the cases, majorly. Then in fungus, it is mostly made up of chitin. In algae, however, we are saying besides cellulose and pectin, other contents can be there, which are galactans, mannans, sometimes mineral compounds like calcium carbonate or silica. Silica can also be there. Where? Diatoms. Calcium carbonate in coralline algae, yeah, those which are making the corals in the sea. And the same way plants are making what? Cellulose, hemicellulose, pectins and proteins. So such type of character we used to mention. So the plants, algae, fungi, bacteria, they have the cell wall, and here are the chemical composition of these cell walls, right? So that's the thing that we can mention over here. Finally, the next thing that we need to mention is, the other thing that we need to mention about is about the cell wall structure. The structure that we are going to discuss here is of plant cell. We are going to study it in the plant cell, okay? Next, the other characters. So what are the parts of the plant cell wall? The plant cell wall is consisting of one, the outermost layer, which is called as middle lamella. Outermost, I said what? Outermost. The second one, obviously inner to the middle lamella, is primary wall. You can say it is in middle. Clear? The middle lamella is actually between adjacent cell. By saying middle lamella means between adjacent cells. So it is a layer of the cell wall which is actually shared between the adjacent cell. Next is primary wall, the primary wall, and then comes the innermost, which is secondary. Now what we are observing here, as per this diagram, the thickest one is secondary wall. The middle lamella is a glue between adjacent cells and having the calcium pectate, magnesium pectate like of compounds, out of which the calcium pectate is more in amount as compared to magnesium pectate. Nextly, the primary wall, which is made up of cellulose type of thing. The cellulose is going to form this structure in the form of tiny fibrils. This is exclusive to a cell, and however, if the secondary wall get formed in a cell, in that cases the primary wall is becoming less prominent, or you can say less distinguishable. The secondary wall is thickest, multi-layered and often with pits. It is the innermost layer, and either suberin or lignin is forming this secondary wall. Remember, when the secondary wall is formed, the content is lost, and we call this empty place as lumen. I am saying protoplast is lost with the formation of secondary wall. So what I have learned from the diagram is the cell walls are getting formed towards the center, okay. Keep this thing in mind. We are going to discuss it in the later topics here itself in the cell wall only. So these are the three different layers along with their chemical composition, out of which the primary wall and secondary wall are exclusive for the cell. The middle lamella is shared one with the adjacent one. One fundamental type of question. See, this is 1, this is 2, this is 3, this is 4, this is 5, and here continuous, so many. This I am saying this is the tip of root. Definitely this area must not have middle lamella. This side middle lamella present, this side middle lamella, this side middle lamella, this side middle lamella. Here continuity of the cells, that's why here also middle lamella. But the tip is lacking middle lamella. Remember this. Okay. Coming to the next one. The cell wall origin in the growth. How the cell wall originated? Actually, it is like when the plant cells were undergoing division. I'll say mitotic division. This is the cell wall. This is the plasma membrane. And then importantly, because the cells are dividing, daughter nuclei, okay, fine. Other characters, what else I need to tell? What is left actually to be told? And that is how this partition is formed. Now this partition is actually going in this way. Earlier to this, some granules were getting aggregated towards the periphery, which granules are random here. So we say that this structure is actually phargoplast, which is formed as cell plate. The cell plate is growing towards the periphery, and this cell wall resulted in the formation of middle lamella, or you can say cell wall. And see here, the cells are interconnected. I will say this structure as plasmodesmata again. Okay, so the origin of the cell wall is from the cell plate, or you can say more earlier as phargoplast. So that during cell division from phargoplast, we are saying the cell plate is formed. The next is formation of plate is centrifugal. Getting my point? See, it is going towards outer side. See the direction, in which direction this cell plate is growing. This cell plate is growing towards the periphery, Okay, so that kind of condition we can mention is the easiest one. Formation of cell plate is centrifugal. Nextly, the growth. What about the growth? The growth of the cell wall means new layers are getting formed, and they are formed towards the center, right? The growth is towards the center, means if I am going to make the cell wall, in that cases it is going to appear in the manner which is 3D. And see how the cell walls are, how the layers of the cell wall are forming towards the center. It is like this. So this is what I am making is middle lamella right now. Right now it is middle lamella. Okay. This is middle lamella. Next is the second layer of the cell wall. Inner to the middle lamella formed is primary wall. This is primary wall. This is primary wall. And inner to primary wall, if further thickening of the wall is taking place or the cell wall grows further, it will be secondary wall. First layer of the secondary wall. Remember, secondary wall is multi-layered. Secondary wall is multi-layered. Remember this condition. See, so many layers are getting formed, and here we are forming number of layers, right? So these are how many? 3. I will write them as S1, S2, S3, together forming the secondary wall. The appearance or the ambience is 3D, 3-dimensional, because the units or the structures, fibrils, are going to arrange in different planes, zigzag, this side, this side, this side, that side. So this is giving the 3D ambience. The structure is appearing 3-dimensional structure. So this is how we can mention the things like this. So that's why we are saying origin is centrifugal and growth of the cell wall is centripetal. One is centrifugal, other is centripetal, right? This is how we can mention about origin and growth. Next. So what the cell wall can perform? The usual comparison is for the bacteria and the plant cell. In the bacterial cell that we have studied was, besides the maintenance of the shape, we have studied this thing, we have been told that beside the structural support to the cell or the shape of the cell wall is protecting the cell to get shrink or to get bursted in the medium. Okay, the cell is not getting collapsed, not getting bursted because of change in the medium. In the plant cell, the cell wall again provides the shape. Nextly, we are saying this cell wall is providing interaction between the cells, plasmodesmata. Clear? The cell wall is holding the adjacent cell, middle lamella. Okay, and then we say that the cell wall is therefore becoming important by disallowing or discouraging the macromolecules to cross through it. Because of the middle lamella components like calcium pactate or magnesium pactate, it is very difficult for the pathogens to penetrate the cell. They can penetrate easily the dividing cells. But when we are saying the cell is matured, they have thicker middle lamella, they have more of the calcium pactate, but as the cell dies or going towards aging, the calcium pactate, magnesium pactate get dissolved, and again the cell is becoming prone for the attack of any pathogen or infectious agent. Just I'll give you the example. When the fruit is ripening, say for example tomato, green in color right now, it will be more tight. Okay, it is going to be more tight, more stiff. But as it is becoming red, and after few days of this redness, we are observing it is becoming soft. So that softness of the fruit is actually because of loss of calcium pactate from the middle lamella. Clear? I can mention this thing very clearly that you need to remember very carefully, and that is what? That softening of ripened fruits. Here what we can say? Senescent cells is due to loss of pactate simply, or just degradation of middle lamella, means the glue between adjacent cells. That's the simple thing. Going for the next to the plant cell. What is next? The thing is, although the word is not common in use, but if there is a question like what about animal cell wall? In that case, the animal cell wall is actually the carbohydrate compositions that are present outside the plasma membrane or the sugar residues of the plasma membrane, which are named as glycocalyx. That glycocalyx is often stated as animal cell wall, and we have studied the function of that structure. It is providing the basis for cell recognition mechanism, that is immunity, antigen antibody specificity, or rejection of transplant organ, etc., etc. Okay, so it is not a common word, remember. It is just on the basis of a question if asked that what is the function of animal cell wall. Don't give a second thought that the cell wall is not there in the animal cell. They must be asking about glycocalyx, right? Next, there are so many cell organelles in the eukaryotic cell. As we have studied, eukaryotic cell means which is having division of labor within the cell, or the cell organelles which with designated functions. But certain cell organelles perform the function in a coordinated way. They constitute the system. It is just like you can take the example of our body, the organ system level, that we are chewing the food, mechanical digestion, partially chemical digestion due to the presence of salivary amylase, the carbohydrate is getting digested. In our stomach, the protein is digested at large, then to the intestine and the other things. Fine. So, for example, some person got peptic ulcer or something, or because of certain surgical reasons, part of the stomach got removed, or you can say most of the part of the stomach got removed and only small part is left. In that cases, what will happen to the digestive activity of that individual? That patient is obviously not supposed to take high protein content diet. Why? Because the major site of protein digestion is not functioning or is lacking. So that's why the system get affected. This is how we say the system is having coordination of different parts. The same is about the cell organelles which perform function in coordination. So this is how the cell organelles, being their function, are coordinated together, are forming the endomembrane system. I am not saying endoskeleton or cytoskeleton, I am saying endomembrane system. And this endomembrane system is comprising of the organelles, namely endoplasmic reticulum, Golgi apparatus, lysosome and vacuoles. Okay, these are the four components. Many of the time, many people used to say GERL as an abbreviation. What is GERL? G for Golgi apparatus, E for endoplasmic reticulum, L for lysosomes. Okay, so these are the things which are constituting the endomembrane system. Now, how they are coordinated? Let me tell you this, a very brief description, an easy one. This is outer nuclear membrane. This one is inner, and now I am making the outer nuclear membrane. This one is outer nuclear membrane. The outer nuclear membrane near the nuclear pore is forming a structure. Means extension of the outer nuclear membrane like this, and taking the cytoplasm into its arms or within the limitations, inside lumen, outside extra lumen. Okay, this way. Now, this structure is called as endoplasmic reticulum. It is often having some studded appearance, or you can say something present as grains on their surface like outer nuclear membrane. These things. Now, these granular particles are actually ribosomes, ATS ribosomes. Because of these ribosomes, the structure is appearing rough. The diagram is inspired from the diagram that is given in the NCERT figure. Another structure can be there. Here I am making, but it is smooth in appearance. It is not having ribosomes on the surface. So, they used to call the earlier one, the former one, as rough endoplasmic reticulum, and this one is smooth endoplasmic reticulum. This inside is nucleus. This one is outer nuclear membrane. These green bodies are ATS ribosomes. The ER has created two places, one inside lumen, outside extra lumen. This place as luminal space and the outside as extra luminal space. Okay, these are the different things. Remember, endoplasmic reticulum separated cytoplasm into two parts, luminal and extraluminal. Endoplasmic reticulum is two types, RER, SER. But from where this ER is originating? I said nucleus. So if nucleus is not there, ER will not be there. In a dividing cell where nucleus disappear, endoplasmic reticulum disappear. Secondly, there are certain vesicles which are coming out from the ER. Okay, and these vesicles are getting fused at a place. These all vesicles are getting fused. These are vesicles, and they are getting fused to form the next structure that is called as Golgi apparatus. That structure is named as Golgi apparatus. So here is the formation of Golgi apparatus. This is the surface to which these vesicles are getting fused, which appears convex on this side. And then nextly, we are saying the same structure getting elongated because of deposition of more number of vesicles, and thereafter it is giving off secretory vesicles from the surface and getting disappeared. These flat sac-like structure are called as cisternae. These are cisternae, right? And cisternae are getting lost. This side is convex, then this side will be concave. So we are observing what happened actually. So the Golgi apparatus is visible when ER is there. If ER is not there, Golgi apparatus is not there. Who give rise to the Golgi apparatus? Endoplasmic reticulum. It is something which is formed on ER, gets stored or modified in Golgi apparatus. This structure is Golgi apparatus or Golgi complex. Now, may be possible that these vesicles which are going out from the Golgi apparatus are going to form lysosomes. Okay, maybe these are going to be lysosomes. That's it. Their formation is obviously related to one another. Their functions are going to be related to one another. The enzymes of lysosomes are formed on ER. Lysosomes themselves are getting formed at Golgi. The enzymes of lysosome formed in ER get packed in Golgi apparatus, and this is how the lysosome came in existence. So their functions are obviously in coordination, definitely. It is clear-cut thing to establish the coordination among the cell organelles, but not all. Mitochondria and plastids are also membrane-bound cell organelles. Mitochondria, chloroplast and peroxisome are the three cell organelles that participate together in one function called as photorespiration in certain plants. That photorespiration involves all three cell organelles. What are they? One is called as mitochondria, second is chloroplast, and third is peroxisome. But still they are not forming the system. Say for example, in my body mitochondria are present, peroxisomes are present, chloroplast is not there. Here their occurrences also depend on one another. You can simply say that mitochondria, chloroplast, peroxisome are not forming any system. Okay, they need to be coordinated in the functions, then only they can perform. To get together or to get associated to perform one or two function doesn't mean forming the system. Okay. The endoplasmic reticulum, Golgi complex, lysosome and vacuoles, these all are single membrane bound organelles. These all are single membrane bound. Double membrane bound cell organelles, mitochondria, plastids are not forming the endomembrane system. Remember what I said. Single membrane bound cell organelles like endoplasmic reticulum, Golgi apparatus, lysosome, vacuoles form the endomembrane system, but the double membrane bound cell organelles, mitochondria and chloroplast are not forming the endomembrane system. So be clear about this. Okay, be sure about these things, right? That which type of cell organelles form the membranes, endomembrane system. Single membrane bound or double membrane bound? Answer is single membrane bound. Okay. Nextly, that endomembrane system, the ER, the endoplasmic reticulum, name says endoplasmic, means inside. Reticulum network is actually found in which places in eukaryotic cell, particularly those which are matured but disappear in the dividing cells. However, if I am saying about human RBCs, those which not have the nucleus, definitely they don't have endoplasmic reticulum as well. They don't have Golgi apparatus also, right? Then about the origin, from where they originated. So I can say that endoplasmic reticulum seems to be the extension of outer nuclear membrane. These extensions which are going to separate the cytoplasm into two parts, extensions. Which separate the cytoplasm into two parts. And I told you the parts are luminal and extraluminal, right? Next comes about the structure. Okay, the two parts, remember what? Luminal and extraluminal. So here is the thing about the structure that I can mention, that the structural elements of ER are, means endoplasmic reticulum, structural elements. What are the structural elements? Means the components. One is cisterna, second is vesicle, third are tubules. Now cisterna are something which are bag-like structures or the flattened sac-like structure which bear ribosomes on the surface. Vesicle, rounded oval bodies which may not have ribosomes on them, whereas tubules are elongated and the branched but smooth. They don't have ribosomes. Okay. So here is the thing that we can mention very clearly, right? So this is about the components of endoplasmic reticulum. Next, the thing about the structure that I told you. So I have two type of things. One is rough endoplasmic reticulum and the other is smooth endoplasmic reticulum. Smooth as what? Smooth as plasma membrane. Okay, rough endoplasmic reticulum as outer nuclear membrane, bear ribosomes on the surface. Which ribosomes? ATS ribosomes. Therefore, granular in appearance. This is granular in appearance. Okay, that kind of character we can mention. And about this smoothest plasma membrane, what about smoothest plasma membrane in this case? The smoothest plasma membrane means they are not granular. I can call them as agranular. Comparison. Okay, comparison. Remember, ribosomes are also named as ergosomes. Fine. These which bear the ribosome have more numbers of cisternae in them. These which are smooth in appearance, the SCR, is having more numbers of tubules in them. Right? These type of things we can mention about endoplasmic reticulum, the types. Okay, the types. So the types 1, rough endoplasmic reticulum, type 2, smooth endoplasmic reticulum. The rough endoplasmic reticulum bear ribosome on the surface, so they seems to be the prominent in the cells which are performing protein synthesis and secretion, whereas the smooth endoplasmic reticulum is the site of lipid synthesis, and in animal cell it is the place of synthesis of steroids. It is not about their function, right? We will study the function separately. In rough endoplasmic reticulum, because ergosome or ribosomes are present, there was the name, the earlier given name was ergastoplasm. This was the earlier given name to endoplasmic reticulum, later came out to be rough endoplasmic reticulum. The same way, the smooth endoplasmic reticulum is also having one more name, particularly in the muscle cell, where these are associated with release and the uptake of calcium ion. I repeat, release and uptake of calcium ion means related to the cell organelle, related to contraction of muscle cell. that is smooth endoplasmic reticulum. Why I am saying this? It is because of the role it is playing regarding calcium ions. Okay, this is about the types of ER. The topic is not going to end here. The endomembrane system, endoplasmic reticulum, more to tell what the functions. The endoplasmic reticulum or the ER is forming the extensive membrane that participate in the membrane flow. It is like if something is made on the nucleus, will first come to the ER is going to pass it to the Golgi, the Golgi is going to pass this thing to plasma membrane, and that may be outside to the cell. This is membrane flow. Okay, they form the endoskeleton, clear, and they are forming the endoskeleton. In that cases, they are actually the membranes. They are actually certain type of wall. I am using the word. They are membranes, they are wall, and therefore they are making the boundaries. They are making the limitations by which different structures in the cell can be kept in proper position, keep organelles in place. And as I told you about plasmodesmata, the protoplasmic bridges that are crossing the cell wall. Remember which one? Middle lamella and primary wall, not the secondary wall. Remember that one. Okay, secondary wall. The cell who is forming the secondary wall need not to have plasmodesmata in them. Why? Because they are dead. No interaction is required. Okay, so the continuity of the membrane through the plasmodesmata is called as desmotubule. Okay, so here I can mention this thing, that this is what I am making is a cell, the middle lamella. Okay, and then the other structures. What other structures? The primary wall, and then is the continuity of plasma membrane, or you can say membrane just. That's it. This is the thing that we can mention. Okay, and then I am saying this is these white thing, these bridges are what? Plasmodesmata, and the continuity of the membrane through it, the continuity of membrane through this membrane from one cell to the other cell is actually what? Desmotubule. They are nothing, they are modification of endoplasmic reticulum, or you can say the membrane. Which membrane? The plasma membrane. Okay, that is desmotubule. Let me mention this very clearly. This is middle lamella, then the black one is primary wall, no secondary wall is there. Be sure about this. Okay, desmotubule is purple in structure. I am mentioning it by 2. Plasmodesmata I am mentioning by 1. Right? This 1 is plasmodesmata, this 2 is desmotubule. Okay, the other things, let's separate the functions into rough and the smooth endoplasmic reticulum. The rough endoplasmic reticulum is known for the protein synthesis and the pecking, then reorganization of nuclear membrane after the completion of cell division. The same way, production of the enzymes of lysosomes. Lysosomal enzymes are proteins. They are made on rough endoplasmic reticulum by ribosomes. The smooth endoplasmic reticulum is related to the function like lipid synthesis, steroid hormone synthesis. Other function includes absorption and release of calcium ions in the muscle cell, and not at all leaving that one, and that is what? Detoxification or xenobiotics. It will be xenobiotics or detoxification next. So this is how we can mention the things. That's simple one. Okay, this is how we can mention the condition like endomembrane system, the endoplasmic reticulum. Here we are going to conclude. We have studied about the origin, we have studied about the functions, we have studied about the types and the structure. Next to the endoplasmic reticulum is Golgi apparatus. Okay, the Golgi apparatus means the Golgi apparatus is the structure which is going to be present in the eukaryotic cell, particularly the matured one except mammalian RBC. And also we are saying it is scattered in plant and invertebrates. That scattered Golgi apparatus is named as dictyosome. That scattered structure is called as DICTYOSOMES, dictyosomes. Okay, these type of character we can mention about them. Nextly, we have the other characters that include what? The origin. The Golgi apparatus originated from endoplasmic reticulum. This we have discussed earlier. Means in the beginning itself. The vesicles from the endoplasmic reticulum, whether SCR or RER, were getting aggregated to form Golgi apparatus. Okay. Then we are saying the structure is comprising of 4 type of components here. They are namely cisterna, then second comes vesicle, third are tubules, and fourth is vacuole. Right? These are the components. Let's see in the diagram where are these things given or how these things can be labeled. Okay. Remember, the Golgi is going to change its appearance. The one who changes its appearance is pleuromorphic. I said appearance is changed. Pleuromorphic, the function remains the same. The storage, modification and secretion. Okay. So here we say what about the polymorphism. So see this, in NCRT this one is actually what? This is the place which is matured side. And this is actually which is going to be formed, means forming side. Wait, the formation is towards the ER or towards the nucleus actually. So here I am going to tell what happened actually and how this structure has been formed. The vesicles were coming from ER. Vesicles from ER. Okay, they were getting aggregated on this surface. When these things are getting merged completely, this structure will become more flattened and more longer. Not only one, several layers will be this. So here I am saying this structure is called as cisterna. And about cisternae we used to mention these are functional unit of Golgi apparatus. These are functional unit of Golgi apparatus. So further, what happened to this cisterna? This is the forming phase. Remember, the appearance of the forming phase is, this is forming phase, or you can say cis phase, which appears convex from the nucleus side. I will say this is proximal, being nuclear side, and this is toward the cytoplasm, means the distal phase or the distal side. Okay, on this proximal side it is appearing convex, fine, and on this side, the distal side, it is appearing, say here I will say maturing. So cisternae are getting formed on cis phase. I repeat, cisternae are formed on cis phase, and on trans phase they are going to give out the vesicles again, secretory vesicles, and they are getting finished. See the difference in the bulgings. Okay, here the bulges are coming into the cisternae, here they are going out from the cisternae, out, gone, out, gone, leaving behind the empty space, leaving behind the empty space. The extensions which are formed because of pinching of cisternae are called as tubules, and the empty spaces are called as vacuoles. These are tubules. It is possible that the tubules may intersect each other, or because of which the structure Golgi apparatus is appearing reticulate. Clear? I have observed this structure Golgi apparatus under microscope using methylene blue. What to use Golgi apparatus? use methylene blue under microscope to see this structure, right? Remember this. Use methylene blue to see this structure under microscope. These are what? These are vesicles. So, maturing phase is the one which is, if that one is cis, this will be trans. Not phase, face. And from the surface it is appearing concave. Wait, not only concave, if it is having some holes in it, just like coffee mug, the froth is appearing with several holes or the network. Fenestrated or reticulate, right? This is fenestrated or reticulate. This is how we can mention this thing. The simple and the easiest one. Remember all the features. This is how the shape is getting changed. So here I can mention the golden lines about Golgi. Cisternae are formed at cis phase and lost at trans phase. Just simple and easier one, right? So this is the structure comparison that we have studied. Next, about the Golgi functions. What Golgi is going to perform. As I told you, it is having the material that is synthesized on the RER and SER. So it means it is storing, modifying the material to be released in the cell or outside the cell. And then glycosylation of protein, glycosidation of lipids. Now the two things are possible because of the particular enzyme. By the enzyme named glycosyl transferase. It is because of the enzyme that is glycosyl transferase. So when we are saying glycosyl transferase, then cases we can say by the enzyme glycosyl transferase, glycosylation, glycosidation means converting protein into glycoprotein, lipids into glycolipids. That's the same one, okay? And then repairing of plasma membrane, or you can say regeneration of plasma membrane. If there is any type of losses or any puncture or any holes in the plasma membrane, they will be repaired by Golgi apparatus. It is responsible for the origin of cell organelles like lysosomes, right? And they are forming the component of membrane flow. Now I can mention the membrane flow in much detailed way. From the nucleus to ER, from the ER to cis Golgi, then I will write median Golgi, and then I will write trans Golgi, and here is the end, right? Going to plasma membrane. This is the membrane flow, extended, okay? So these are the functions of Golgi apparatus. There are certain modifications like acrosome, cap of human sperm, zymogen granules are obviously what? These both are modified Golgi apparatus, right? This is Golgi apparatus. Done. Next to the Golgi apparatus was just I said that this is going to store, modify the materials. In that cases, it can be mentioned as warehouse of the cell. Some people mention, you can find it in the old books, traffic police of the cell, which is regulating the secretion from the cell actually. Okay, that's it. Coming to the next, lysosomes. Now I have directly mentioned that this is polymorphic cell organelle because there is nothing much to study about the structure of the lysosome and their occurrence. The lysosomes are present in the cells which are particularly performing intracellular digestion. Now when the material is getting intracellularly digested, many of the compounds get digested in the cytoplasm, which is alkaline medium. Okay, cytoplasm which is alkaline medium. Let me make a cell here, then we can mention the things very clearly. So this is a cell. I am saying this one is cytoplasm, right? This is cytoplasm, and the pH value is alkaline. Okay, so different enzymes will be there in the cytoplasm to perform the breakdown of carbohydrate amylases, to break down the compounds like nucleic acid nucleases, to break down lipids lipases, to break down proteins proteases, but they all are acting on alkaline medium. Okay, if the medium get changed, those enzymes become non-functional. Then why lysosomes are there? It is because if the material is not getting broken down in the cytoplasm, that material is going to be sent to, we are going to send this material to the structure which is called as lysosome. Okay, if anything, whether it's any expired cell organelle, if it is any expired cell organelle, means the function is over, the turnover number is over. Or maybe the particle which is getting engulfed from outside, engulfed food particles, or it may be certain antigens also which invaded the cell, either antigens or you can say allergens which invaded the cell. Now this is the lysosome. I am saying this is primary lysosome. And be clear about this, I said polymorphic, not pleuromorphic, the one who is going to change its function with the time. It is going to change its function. The primary lysosome is present. Who is having what? Inactive enzymes right now. But remember about these enzymes. These enzymes which are present in the primary lysosome, are going to work in acidic pH. Got it? If a material is not getting digested in the alkaline medium, will be sent to the acidic medium. The enzymes in total are called as acid hydrolases. Why? Because these all are hydrolytic enzymes, means they break bonds using water. Okay, so they are acid hydrolases. Enzymes are inactive. So the question is, what will happen if the enzyme of lysosome get leaked? Few of the enzymes of lysosome get leaked in this cytoplasm. Answer, nothing. Why? Because the alkaline medium is there, whereas the lysosomal enzyme can function well in acidic medium. So release or the leakage of few enzymes from the lysosome are not going to cause any harm to the cell. Not any harm to the cell. Okay, so here we say for example the engulfed particle are coming to it. If it is so, in that cases the phenomena is mentioned as, that phenomena of taking the particle from outside is called as heterophagy. This phenomena is called as heterophagy, or it may be going to take up the expired cell organelle, then I will call it as autophagy. Okay, autophagy means a good thing. Why? Because this lysosome is cleaning up the cell, it is removing the waste. So here we say cell scavenging. Okay, this is cell scavenging. Now the primary lysosomal enzymes become activated, and now they are going to digest the particles which they have engulfed from the outside or from the inside, maybe expired cell organelle or any macromolecule, okay, which is required to be recycled. Now the digestion is going to take place, and we call this thing as, now the next structure is called as the secondary lysosome, where the digestion took place. The secondary lysosome. The vacuole that has been formed because of heterophagy is heterophagosome. Because of autophagy is autophagosome. They get fused with the primary lysosome. Digestion got started. This is secondary lysosome. Now this secondary lysosome is going to perform what function? Digestion. Digestion is over. The nutrients are getting absorbed, right? The nutrients are getting absorbed. Absorption of nutrients which are digested. Absorption of digested nutrients. And thereafter it is about this structure, means which is not at all required now. It is tertiary lysosome. This is tertiary lysosome, also called as residual body. Means it is going to remove the structure which are of no use. I can say these all are waste or to be rejected from the cell. So it thrown out the material. This phenomena is called ephagy or cell vomiting. So we have observed that there are three different roles of lysosomes. One, primary, having the enzymes only. Second, secondary, having digestion as the function. Third, tertiary lysosome, which is going to get rid of the waste material, right? That is why we are saying lysosome as polymorphic cell organelle. So I told you if few enzymes are getting leaked, there is no problem. But if many enzymes are leaked, then it will result in autolysis. Now what autolysis means? Clear? Leakage of few enzymes is not going to affect. Why? Because the medium of the cytoplasm is alkaline. But if so many enzymes are leaked, then it is going to cause autolysis. What does it mean? This is equivalent to cell suicide. Why? Because in these cases, the pH of medium or the cytoplasm get altered. And enzyme of lysosome begin to consume the cellular content. Which is useful also. Now, consuming the cell content is not autophagy here. It is going to be something disastrous, going to consume the useful thing also. That's why it is related as cell suicide. It is not good. An example of autolysis, yeah, disappearance of the tail of tadpole when it is getting metamorphosed into frog is an example of autolysis. Okay, right? Digesting up all the things of the cell. The cell suicide. Lysosomes are called as suicidal bags therefore. Rest of the functions I told you, they are going to recycle the macromolecules. They are going to make the cell clean or neat or doing cell scavenging. This is how the lysosomes are important, right? The next thing about the endomembrane system is about the vacuole. Now the vacuole are the membrane bound space found in the cytoplasm. They contain water, sap, excretory product which are not required in the cell anymore, or maybe a waste, not a metabolically active portion of the cell. This cell wall, this cover of the vacuole is called as tonoplast. Okay. So vacuole, where it is found and what they are doing. If I am going to study about the plant cells, in plant cells sap vacuoles are present. Sap vacuoles. Those sap vacuoles contain the sap which is having high concentration as compared to the cytoplasm. Because of the high concentration, we are saying this sap vacuole is responsible for the cell shape and turgidity. Okay, it is maintaining the cell shape of the plant cell. I can retain these things. How? That membrane is allowing uptake of ions against the concentration gradient. Against the concentration. Okay, means actively with the expenditure of energy. This is why we are saying the sap is having higher concentration as compared to the cell. Okay. Next, The freshwater protists, the unicellular eukaryotic one, which are present in the freshwater, they are taking water regularly from their surrounding and becoming, obviously having excess of water in them. To get rid of excess water, they have contractile vacuole. And just now what I said, the engulfment of the food from the outside, right? Heterophagosome, from the inside, autophagosome are the names of food vacuoles. But don't get confused with the structure that we have studied in the prokaryotic cell. That gas vacuole that we have studied in the prokaryote is actually a pseudovacuole. It is not having any membrane. Remember, no limiting membrane. They don't have any tonoplast. They don't have any tonoplast, right? So this is about the vacuole, and this is complete about the endomembrane system. In the coming classes, we are going to study about the structures of the double membrane bound cell organelle and ribosomes. So be regular with the classes, go through the DPPs so that you can increase your efficiency and accuracy. Thank you for watching the classes on Yup Master. All the best.